Okay, so on Wednesday, we talked about chapter 12. Today, we are gonna do most, if not all, of chapter 13. See. Which is again, things that you go covered in lab already, so. Learning all. There's a quiz today. It's open till 11:59 p.m. Do something to remind yourself that you're going to need to do that. This wants to keep opening. I've closed it 100 times. I don't know what it's going on. I know, we may be spending our whole class period closing that. I don't know. I minimized it. I closed it. I minimized it. I closed it. It's not coming up in Task Manager. Anyone have any ideas?
Have you tried turning the computer off and back
That
on?
is always a good suggestion. No, I have not tried turning the computer off yet, but that may be what we get to. Chapter 13 and 14 homework said it will be due on Monday. It's gonna be due on Wednesday. It'll be posted later today. I need to answer some more questions. So it will be up there, as you can see. There's a nice reminder of that. Your next exam is gonna be the week of November 11th. Which is not yet here, but I talked to Dr. Dietrich yesterday, and we are not lining up, so we're not worrying about it. I think you guys have another term exam next week, or the week after. Oh, the retest is next week. And our exam will be the week after. So. Questions about schedule stuff? I feel like we are very sparse today. Okay, questions from last class. Take a minute to look at these, and then I might restart the computer and try this again. Okay, I want us to get back into the mentality of mitosis because mitosis and meiosis have similarities and differences. So we're going to get back into that mentality a little bit with these questions that you can't see. Let's try and do this. Okay, so the first question says, what are the phases of the cell cycle? Think about that for a second while I try re-logging in. So you should have 7-ish, depending on how you break it down. Okay, we'll see if this works. Okay, what are the phases of the cell cycle? Interphase. Prophase. Prometaphase. Metaphase. Anaphase. Telophase. Cytokinesis. Okay, when in this process is it replicated? When is it copied? During which of those steps? During S, which is part of interphase. So interphase breaks down into 3 stages, G1, S, and G2. So S is when the DNA is replicated. If you start before mitosis with a cell that has 18 chromosomes, how many daughter cells will there be at the end of mitosis? 2, because it's in the name, mitosis. Okay, it says 2 in the middle, TO. How many chromosomes will each one of them have? 18, because we are making an exact copy of that cell. So if we start with 18 chromosomes, we end up with 18 chromosomes. If we start with 46, we end up with 46. If we start with 197, we're gonna end up with 197. There are no cells with 197 chromosomes left. Okay, it's an exact copy. So we're gonna get into meiosis today, which is not an exact copy. When we talk about meiosis, we're talking about the life cycle of sex cells. So things that go on in my ovaries and testes. So I think of it as like mitosis happens in my toes because it's happening in all the somatic cells, and meiosis, this doesn't work for everyone, sorry, happens in my ovaries. Okay? So mitosis is in your toes, your somatic cells, meiosis is happening in the ovaries for the testes to produce sex cells. So when we talk about this, this is the beginning of our unit that's on genetics. So genetics, when we talk about genetics, we're talking about heredity, traits being passed on. And variations that are inherited. So we're starting with the very foundations of how do we make gametes so that we can make offspring. And how does that result in genetic variation? And then we're going to go into all the fun of Punnett squares. And dihybrid crosses and monohybrid crosses as we get into chapter 14. So the hereditary units that we're going to be talking about in this unit are genes. So when we talk about genes, we're talking about a section of DNA. We talked about those long linear chromosomes that you have, 46 of them. When we talk about a gene, we're talking about one section of that. So that is a section of DNA that's going to code for a specific trait. We don't really know how many genes we have, but somewhere around 20,000 in humans. So in humans, we're talking about 46 chromosomes. Each chromosome has somewhere from several hundred to, oh, that doesn't say 100, that's an arrow, to several thousand genes on it. So we're going to be thinking mostly in this chapter about the idea of chromosomes. So we're talking about a unit of DNA that has a bunch of genes on it between several hundred and several thousand. Someday you should come visit me in my office. I'm outside my office door. There are all 23 chromosomes of the human body in there. It shows you where the genes are for all different kinds of genetic disorders. All kinds of very interesting kinds of things. Mobius syndromes. What is it? I don't remember. We looked it up. Okay, so all of these genes are carried on these chromosomes. Today, we're going to be talking about meiosis and how we split those up. So talking about the idea of inheritance, we're going to talk about the two kinds of reproduction at a very, very basic level. There are two kinds. Actually, I think it comes up. Yeah, I don't need to write. There's two kinds of reproduction that can occur. There's asexual reproduction. How many parents are involved in asexual reproduction? One parent. Asexual means there is no sex involved. So this is one parent. We're going to produce clones. This produces clones or exact copies. So this is what we're talking about when we think about mitosis. Mitosis can be a form of asexual reproduction. There are other forms of asexual reproduction, like this spider plant. Okay, this spider plant produces these little offsprings. Those are clones. You can take those off. That's called vegetative reproduction, but it's still asexual reproduction. Another example is Hydra, which are part of the Cnidarian family, like jellyfish. They can bud, so they can just grow off a little baby off the side, and then it pops off and becomes a new offspring that's a clone. So when we talk about it, we're not just talking about the process of mitosis. There's lots of different forms of asexual reproduction. In Bio 112, sorry, 1120, we will talk about lots of different life cycles and ways that organisms can reproduce asexually versus sexual reproduction. There are some advantages to asexual reproduction. If you've got good genes, you can pass them on. It takes a lot less time and energy because there's only one parent involved. It doesn't involve a lot of finding a mate, making sure you're pollinated, none of that has to happen. So there are some advantages of asexual reproduction. The opposite of that is sexual reproduction. That involves how many parents? 2. Because of that, because there are 2 parents, you're getting some kind of combination of genes. What does that mean for your offspring? Compared to the parent, they're different from the parent, and compared to each other, usually they are different. If any of you are identical twins, it's not true, but this results in genetic variation. Offspring are different from each other. Offspring are different from the parents. This has some advantages. What are some of the advantages of this? Survival. Yeah, you can have offspring that are different from you. So if you have a trait that is not beneficial, your offspring may not have that trait. They might have a different trait that is beneficial. So it's the ability to pass on this genetic variation can create a stronger organism. And then that organism can pass on that trait to their offspring. So genetic variation in general is a benefit. What are some disadvantages of sexual reproduction? It takes more time, it takes more energy. If you're a plant and you want to reproduce sexually, generally that means you're making a flower, and it takes a lot of energy to make a flower. And even if you make a flower, it doesn't mean your flower is even going to get pollinated. So there's not even a guarantee that you made this beautiful flower and you're going to get a seed that is genetically different than you. So there's not a guarantee of anything. It takes more energy, it takes more time, but you get genetic variation. Brandon, did you have a question? Okay. So how do we get this genetic variation from meiosis? Is what we're going to be spending the rest of today and maybe a tiny bit of Monday. We'll see. But we're going to review some vocabulary first. Okay, remember, we talked about mitosis on Wednesday, and I brought the reads today. So this is a chromosome. When is it going to replicate? Interphase synthesis. It's going to replicate. Now I have how many chromatids? 2. So I have 2 sister chromatids. They're connected here at the centromere. Still counts as 1 chromosome, but now it's 2 chromatids. So sister chromatids are duplicated chromosomes. From the S phase. We talked about that already. What we're going to introduce today, and what you have seen in lab this week, is that not only are we talk about sister chromatids, we also talk about homologous pairs. Okay, so you have 46 chromosomes, but they line up as 23 pairs because one of those chromosomes came from your mom and one came from your dad. So these are carrying the same genes, not necessarily the same genes. They're carrying the same alleles, the same information. They're carrying information about the same traits, not necessarily the same information, but they're carrying information about the same traits. So they're considered homologs or homologous because they're carrying the same kind of information. So maybe this is the gene for tongue rolling, which actually is a genetic that we'll talk about. Okay, so maybe this is the gene for tongue rolling for mom, and this is the gene for tongue rolling for dad. And maybe you have the dominant gene, the recessive form of the gene, but they're both the same. It's the same gene, you just might have different forms. So when we talk about homologous chromosomes, we're talking about a pair of chromosomes with the same information or the same genes, one from the male, one from the female. So these are 2 totally separate chromosomes. They are never going to attach to each other. They will never be sister chromatids. So this is showing you here a pair of homologous chromosomes. I did not put I put this too close to the top. So this is a pair of homologous chromosomes. These are duplicated. So they have sister chromatids. So the sister chromatids are connected at the centromere. The homologous chromosomes are not connected to each other directly. So this is just showing you this is a duplicated chromosome. Over here, what you see, this is what we call a karyotype. This is all the chromosomes from a human being. So there's 23 pairs. So numbers 1, I'm just gonna write it below. Numbers 1 through 22 are what we call the autosomal chromosomes. Chromosomes. Sometimes I write the word too many times. Pair 23 are the sex chromosomes. These are the X's and the Y's that are there. And you can see there are different sizes of chromosomes. Some of them have more genes than others, though they generally are arranged from smallest to largest. Sorry, largest to smallest. I do know the difference there. But number 1 is largest and number 22 is the smallest. So when we talk about cells, what we're going to be doing in meiosis is cutting the number of chromosomes in half. So we have to understand the words diploid and haploid. So a diploid cell is a cell that has the full number of chromosomes. So it has a full set. For humans, this is what? How many chromosomes do humans have? 46. So a diploid cell for humans has 46 chromosomes, and we call this, we indicate that as a 2N. So 2N just means a diploid cell. In mitosis, you're starting with diploid cells and you're making more diploid cells because you're copying everything exactly. When we get into meiosis, we're going to start with a diploid cell again, but we're going to turn it into a haploid cell, which has half the number of chromosomes. Which it kind of sounds like half, so that's kind of nice. So for humans, that would be how many? Half of 46 is 23. And that's what we call a haploid cell, and we use N to indicate that. So over here in this cell, you can see some of the same things. We've got a chromosome, sister chromatids that are joined at the centromere. We have here a pair of homologous chromosomes. And then we have this, which is a chromosome with sister chromatids. So this is one duplicated chromosome. Hey guys, if you want to talk, do it later, please. It's distracting to me all the way up here, so I'm assuming it's distracting to other people too. In this case, we have this homologous pair. You can see we have a blue one circled and a red one circled. Those are non-sister chromatids. So they're still chromatids, but they are non-sister because they are not duplications of each other, because we're using red to represent ones that came from the mom and blue ones that came from the dad. So the total number of chromosomes in this cell is how many? We've got 6 chromosomes, so the diploid number is 6. Half of them, the haploid number, 3 came from the mother and 3 came from the father. So the haploid number for this cell would be 3 in the end. So that's vocabulary. We're going to do an overview and then we're going to get into the stages. So meiosis is a little bit more complicated than mitosis. Some similarities, some differences. They both start like the cell cycle. This is still a cell cycle kind of process. They're both starting with interphase. And again, that's most of the cell's life is spent in that phase. So these chromosomes are not yet duplicated. but they would be considered homologous. So we're starting with a parent cell that is diploid. How many chromosomes does this one have? 2. Okay, so we're keeping it simple. Imagine in your cell there are 46. This one we're dealing with only has 2. So these chromosomes are going to duplicate again. That's in the S phase of interphase. So now I have one homologous pair of duplicated chromosomes. And each one of those chromosomes is duplicated, so it has 2 sister chromatids. How many chromosomes total are in this cell? 2. We still just have 2 chromosomes. They're just duplicated now. We have 4 chromatids, but 2 chromosomes. So we enter meiosis 1. We're starting with 2 chromosomes. At the end of meiosis 1, this is what our cells look like. How many chromosomes are in each of those cells? How many chromosomes does this cell have? Hesitantly, 2 people say 1. Great. It has 1 chromosome. This cell has 1 chromosome. So we have cut the number of chromosome cells in half. So in meiosis 1, we're taking 2 end cells, we're taking diploid cells, and we're turning them into haploid cells. We're splitting the homologous pairs. So that's what you need to remember about meiosis 1. The point of meiosis 1 is to split a homologous pair. So if these are our chromosomes, so these are our chromosomes in interphase. I have 2 chromosomes. I'm going to duplicate them. Now I have 2 chromosomes that are duplicated. They're going to line up together because they're homologous. And in meiosis 1, I'm going to pull them apart. Okay, now I have 2 cells. Each of the 2 cells has 1 chromosome because I split this pair in half. From this point on, it's going to look like mitosis. We're going to take those haploid cells and we're going to split them into more haploid cells, but we're going to separate the sister chromatids. So I had 2 haploid cells. Now I'm going to have 4 haploid cells. So meiosis 1 separates the homologous pairs. Meiosis 2 is then going to separate the sister chromatids. So I started with 1 cell that had how many chromosomes? Here I have 1 cell with how many chromosomes? 2. Now I have 1, 2, 3, 4 cells with 1. So I cut the number in half. And I got 4 cells out of a 2. So we're going to talk about how this happens. And you're going to see, again, a lot of similarities to mitosis, especially when we get to meiosis 2. So meiosis 1, the point is to take the homologous pairs to separate them. We're taking a diploid cell, making a haploid cell. So in our cell over here, we've got a pair of homologous chromosomes. I'm going to say it over and over and over and over until you're tired of hearing it. Maybe you already are. These are just the sister chromatids. So these are duplicated chromosomes. And as we go into prophase 1, we're gonna see several things happening. Okay, some of them are the same as what we see in mitosis. The spindles are forming. The chromosomes, the centrosomes are forming. They're moving to the poles. Let's just try that again. So spindles form, the centrosomes move to poles. The nuclear envelope's going to go away. And that DNA is going to condense. So again, it's going to go from being this kind of relaxed state where you don't see it so much looking stringy in the nucleus to where it's condensed and it looks stringy in the nucleus. All of that is pretty much the same as what happens in mitosis, but there's a couple important differences. Oh, sorry, the kinetochores are also going to attach. The big thing, let's make it a different color because it's exciting. The big thing that's happening in prophase 1 that's really important for this whole process is that we get crossing over. So as these chromosomes are lining up, and I know you did this in lab, as these pairs of chromosomes are lining up, so I have my duplicated chromosomes, as they line up, there are enzymes that can work much more elegantly than I can. So that arms, part of the arm of the non-sister chromatids that are next to each other can actually flip. And that place where that occurs is called the chiasmata. So we have this switching of DNA, and that's really important for what we're going to talk about with genetic variation. So this happens on average 1 to 3 times per pair of chromosomes in your cells. So again, we're modeling this with like 2 pairs of chromosomes. Imagine there are 46 pairs of chromosomes. Sometimes they cross over once, sometimes they cross over twice, sometimes they cross over 3 times. It varies every time. And we're going to see what the effects of that are. So this crossing over is an exchange of genetic information between those non-sister chromatids. If it happened between sister chromatids, it wouldn't matter because they have the same information. But here, because one chromosome is coming from the male and one chromosome is coming from the female, and we're switching genetic information, we're producing a unique combination of genes on that chromatid. So these locations where the crossing over happens are called the chiasmata. So at the end of prophase, we have no more nuclear membrane. The homologous pairs are lined up. Crossing over has happened, and it's going to start to look similar to what we saw in metaphase in mitosis. In metaphase 1, those pairs of homologous chromosomes are going to line up in the center at the equator at the metaphase plate. As those spindle fibers are attaching at the kinetochore, this spindle fiber that's connected to this blue one is going to connect to both sister chromatids. This one is connected to both sister chromatids. So as it moves it apart, as we split them apart in anaphase 1, we're no longer pulling sister chromatids apart. We're pulling homologous pairs apart. Which is the point of meiosis 1. Those homologs are going to separate. But the sister chromatids do not separate. So the cohesion proteins that are holding the sister chromatids together at the centromere are still intact. We're just losing the associations that happen between the homologous pairs. So they're going to start to move to the opposite poles. And then we're going to hit telophase 1. Also including cytokinesis in this. So in telophase 1, the cells are going to split. And it's the same thing that homologs. So homologs is just another way to say homologous chromosomes. It's a shortened way. So we don't have to write out the word chromosome again. So these cells are going to split just like we saw before with us. It can be a cleavage furrow or the formation of a cell plate. So the cells split. And we end up with 2 haploid cells with duplicated chromosomes in them. So sometimes at this point, depending on the kind of cell, some of them will actually form a whole new nuclear envelope like they're done. Some cells do not. There's like a gap period, like an interphase period, but there's no more chromosome replication. So we're not going to copy the chromosomes again. Questions about this? Okay, tell someone next to you, what is the point of meiosis 1? What happens in meiosis 1? Better leave you alone. Okay, so now we are going to take, how many cells do we have? 2, and we're going to go through a second round of divisions that's going to look just like mitosis, except for the fact we're starting with a haploid cell. So meiosis 2 is now about the separation of sister chromatids. Because we still have duplicated chromosomes, we're going to split those sister chromatids apart. So we're taking haploid cells and we're making more haploid cells. We're taking duplicated haploid cells and we're making unduplicated haploid cells. And we're going to see the same 4 phases, which are what? Prophase, metaphase, anaphase, telophase. Except this time, it's going to be prophase 2, metaphase 2, anaphase 2, and telophase 2 / cytokinesis. I don't know why we don't label cytokinesis as 1 and 2, but. So prophase 2. There's honestly not anything really exciting that happens. Okay, the spindles are going to form. If you're like me, you might be like, why did the spindles go away? I don't really know. If the cell made new nuclear envelopes, those nuclear envelopes are going to divide. But there's nothing really spectacular going on here. So we'll just skip to metaphase 2. And metaphase 2, we see the chromosomes are lining up at the metaphase plate. So again, they're lining up here, but this time not as pairs. They're lining up in a single line. because there are no pairs anymore. Because the pairs have been split, we're just lining up as single chromosomes that are duplicated. And the kinetochores are going to attach to the different centromeres. Somewhere it's really pro-metaphase. So before the kinetochores were attaching to the centromere, but both chromatids, now they're attaching to the individual sister chromatids. And as we move into anaphase 2, those cohesion proteins that hold the sister chromatids together are going to be pulled apart, just like you pulled the magnets apart in the lab. Those cohesion proteins will break down and the sister chromatids will separate. And this is happening twice because we have 2 cells that we made. So these have lined up in the center. They're going to pull apart. Now I have individual chromatids. And in telophase 2, we are going to get a new nuclear membrane because we're done. So no matter which kind of cell is doing this, the nuclear envelope is going to form again. The chromosomes are going to go from being condensed to being more spread out again. So it's not, the DNA won't look as stringy when you look at it under the microscope. So the DNA is going to decondense and the cytoplasm is going to split. So cytokinesis happens and we're going to end up with four cells. So as you hopefully saw in lab, and you can kind of see in this diagram, how do these cells look compared to each other on the beforehand? Okay, how does this cell look compared to this cell? Are they the same? Are they the same as the cells they started with? So we've created unique cells. They're genetically unique. They're unique from the parent and they're unique from each other. They are not the same as each other and they are not the same as the parent. This is why you don't look just like your siblings, because the eggs that your mom produced were not all identical. And the sperm that your dad produced was not all identical. Okay, if meiosis didn't create any kind of genetic diversity, then all offspring would be identical twins. So this creates genetic diversity. Do some siblings look more alike than others? Yes. But they're still genetically unique unless they are identical. My sister and I are not identical twins, but people still mistake us for twins. We're 3 years apart. I was at a meeting last year and somebody came over to me like, do you teach at Bowdoin? And I was like, no, that's my sister. And I feel like identical copies. And I was like, we're not really, but that's okay. But we look a lot more alike than some other people. You know, people talk about genes being strong, certain genes, you may look more like your mom's side of the family or dad's side of the family. You're still genetically unique. Unless you're an identical twin. Okay, so let's compare mitosis and meiosis that we're going to talk about. We might really get through all this. We're going to talk about genetic variation. I think we're going to. Okay, so mitosis. We're starting with a parent cell every time. We're starting with a parent cell that is diploid or haploid. Diploid. We're starting with a diploid cell. In mitosis, the individual chromosomes line up in metaphase. That doesn't even look like letters. I'm not sure that's any better. The individual chromosomes will line up, and then the sister chromatids separate. And we end up with diploid daughter cells. How many divisions are there in mitosis? 1. Okay, when we go to meiosis, in meiosis 1, in metaphase 1, the homologous pairs line up. And in anaphase, the homologs separate. The homologous pairs separate. So we're starting with a 2N cell and we're ending up with a haploid cell. Then in meiosis 2, those individual chromosomes are going to line up and we're going to end up with more haploid cells, but in this case, it's the sister chromatids separate. So how many divisions are there in meiosis? 2. In mitosis, is there any synapsis of the homologs? Do the homologous pairs line up and exchange genetic information? No. So that is not happening in mitosis. It is happening in meiosis in prophase 1. So synapsis is just when they line up and exchange DNA information. How many daughter cells are produced in mitosis? 2. How many are produced in meiosis? 4. I used to remember that because meiosis has 4 vowels in it. It has E, I, O, I. But it doesn't really work because mitosis has 3 vowels, so that doesn't really help. But if that does help you, it helps me. In terms of genetic composition and mitosis, are they the same or are they different? Same, they're identical. In meiosis, they are unique in relationship to the parents and to the other ones. So lots of similarities, but some really important differences that lead to what we call genetic variation. Genetic variation arises 3 different ways in meiosis. We get genetic variation first, well, not first, one way. In terms of independent assortment. So in metaphase, really, it's metaphase 1. Showing you metaphase 2 here. The homologous pairs can line up in different ways. They can line up independently of each other. So this is showing us 2 possibilities here. We have here where both of the maternal chromosomes are on one side and the paternal chromosomes are on the other side. Or it could happen where you have one maternal chromosome here with a paternal one here, and these are switched around. Where they line up determines how they look at the end. So if they line up this way, we get this cell that has 2 paternal, this cell that has 2 paternal, 2 maternal, 2 maternal chromosomes, whereas this one has 1 of each. they're not gonna line up. When we're talking about 46 chromosomes, you're gonna have them all different which ways. That's a lot of different possibilities for the maternal, paternal, maternal, lots of different ways that they could line up. So that you have lots of different ways for the offspring to look, these daughter cells to look, based on the way those chromosomes line up only. So one way that genetic variation occurs is in the way that the chromosomes line up in pairs in metaphase. One. So that's one way that contributes to genetic diversity. A second way this contributes to genetic diversity, meiosis contributes to genetic diversity, is the crossing over that occurs in prophase 1. So when those homologs line up and exchange genetic information, again, that can happen any number of times. 1 to 3 is the average for a homologous pair in a human chromosome. Sometimes it may not happen. Sometimes it may happen more than that. So you have 23 pairs that can exchange a whole bunch of different ways. That's going to create a whole bunch of diversity. So when they have crossed over, which this doesn't even show, I don't know why. We call them recombinant chromosomes. And that's what you looked at in lab when we were looking at the fungus, the ASCII of the fungus with the different orders of the colors of the spores, the brown and the blacks. You could tell if it was recombinant or non-recombinant. So did any synapses happen? You would see like black, black, brown, brown, black, black, brown, brown. But if it didn't happen, you might see 4 black, 4 brown. So that was creating genetic diversity based on whether or not recombination happened and how it happened. And the third way that this contributes to genetic variation overall is random fertilization. So when you have lots of sperm and lots of different eggs, one sperm fertilizing the egg will produce a different offspring than a different sperm fertilizing the egg. Okay, so fertilization is random, and so you end up with random combinations based on that. And again, why is it important? Why is genetic variation important? Because we can pass on genes that help with survival, and we can create new combinations of genes that are beneficial. So genes that contribute to survival are passed on, and we can get new combinations of genes that are beneficial. Mutations can also happen when this process doesn't happen perfectly, and we'll talk about that a little bit more when we get to DNA replication, but that's also a source of new traits that creates genetic variation, but not really from meiosis. Okay, so this week, I looked at the syllabus and said, no way, but we did. We did 11, we did 12, and we did 13. So on Monday, we will start with chapter 14, which is Mendel and the laws of inheritance and all that. What are you going to do today before 11:59 p.m.? Take a quiz, and you get out a minute early. Happy Friday.